Good afternoon, everyone. We are going to wrap up lecture 2 today and start on number 3. So last class, we talked about lipids and proteins as we continue to discuss the topic of macromolecules, and there are only um, just one more type that we need to cover, and that would be nucleic acid. A nucleic acid. Nucleic acid makes up our DNA, and you've probably heard of DNA before. But there are also something else called RNA that's in our body, and that is also nucleic acid. Now, what you're looking at in this picture are the DNA, the chromosome actually, and they are not naturally colored, but they are able to pick up dyes when we apply certain uh, colors to them, and you can easily line them up in a picture like this. So something like this is called a karyotype, and we'll learn more about it later on in the course. Okay? But it gives you a very nice view of all the chromosomes that we have in the body. We have a total of 23 pairs. The picture is a little bit cut off here, so it stopped at 18, but we do have 23 pairs, 23 from mom and 23 from dad, a total of 46 chromosomes. So let's take a closer look at nucleic acid. The elements that makes up nucleic acid includes carbon, hydrogen, oxygen. There is the triplet again, right? These three things are basically in all the macromolecules that we talked about. In addition, for nucleic acid, we also have nitrogen and phosphorus. The building blocks for nucleic acids are something called nucleotide. Nucleotide, right? So a nucleotide has three parts to it. Okay, I'll write it down for you. A nucleotide has a phosphate. Now you might wonder, like, what's the difference between phosphorus and phosphate? Okay, phosphorus is the element, okay, the element on the periodic table. Phosphate is something you will learn in chemistry later, but it is PO4 -3. Right? It's one phosphorus connected to four oxygen, and it has a charge of -3. So that's what a phosphate is a group of atoms as opposed to phosphorus, which is by itself. So phosphate is going to be connected to some kind of sugar, and then it's going to connect to a base. Okay, we call it a nitrogenous base. Nitrogenous base. Okay, so that's a nucleotide. It has three parts to it. Okay? Just like when we learned amino acid last class, it has different parts to it as well, right? It has the carboxyl group, the amino group, and the side chain. Similarly, a nucleotide, which we don't have to draw, has three parts to it: a phosphate, a sugar, and something called nitrogenous base. Now, there are two main kinds of nucleotides in the body. Two main kinds of nucleic acids in our body. We have DNA, which stands for deoxyribonucleic acid, and we also have RNA. Okay, you might not have heard of RNA before, but during the pandemic, the vaccines, if you received the Pfizer and the Moderna vaccines, for example, they are mRNA vaccines, and the RNA is this RNA. It stands for ribonucleic acid. Okay, so two kinds of nucleic acids in our body. Let's take a closer look at the nucleotide. The nucleotides, again, these are the building blocks, the monomers for nucleic acid. We see the phosphate group here, the PO4. It's connected to a sugar and a nitrogenous base. Now, depending on whether we are talking about DNA or RNA, the kind of sugar that is in the nucleotide will be different. For DNA, the sugar is called deoxyribose. For RNA, the sugar is called ribose. So what's the difference? Okay, so by now you should, have a good understanding that sugar ends with OSE, right? Like glucose, fructose, galactose. So this one is no different. It's a ribose, but on the DNA, if you compare the two structure here, you can see the DNA is missing an oxygen on the sugar. That's why this sugar, even though it's like the ribose, but it's missing an oxygen, so we say deoxy, right? It's same as ribose, but missing an oxygen. Okay. The phosphate groups are the same, and then we have this nitrogenous base. A nitrogenous base. For DNA, the nitrogenous base can be A, C, A, G, C, or T, and I'll explain what those are in a bit. For RNA, it's also AGC, but instead of T, we have something called U. All right, so that's uh, what the nucleotides are. So basically, the DNA has four types of nucleotides, A, G, C, T, whereas RNA, it's A, G, C, or U. Do you guys understand? Right? Just to help you kind of connect this with before, right? Just like amino acid, right? Each amino acid has like a core part, but then, the R groups, what makes them different, right? Different R group, different side chains. And for nucleotides, the core part are the same. What makes them different would be the ACTG, the nitrogenous base. Questions?
Also for DNA, the hydrogen is different.
So
It's
the
here
what
that the yellow, no, green side.
this thing?
That green. So yeah, it
Yeah,
looks
so
different than.
because so three of them are the same, AGC,
Hmm.
AGC, and in this particular picture, they draw the T, right, the thymine, but over here
Hmm.
they drew the uracil, which is what the U stands for. Okay, if they draw A here and A here, then it would have looked the same. All right,
Hmm.
so it's just the
All right.
choice that they make, right? There is another question over here. The main idea is to just understand what difference they have. Yeah, I mean, like the key thing to take away from this particular slide is the difference between DNA and RNA, right? So at the end, we can write a comparison chart, but one of the difference is the kind of sugar you have in the nucleotide, and another difference is the kind of nitrogenous base that is available. Okay, let's move on and see a few more slides. Maybe it'll make more sense. So here are the nitrogenous base, and you can categorize them based on the number of rings you have in the structure. Some of them have two rings, and others they have one ring in it. If it has two rings, then we call it a purine. Purine, okay, two rings in their structure. You don't have to know how to draw them. You don't have to recognize them. I'm not going to show you a picture and ask you what you're looking at, okay? But that's the purine, two rings. And these ones, the pyrimidine, they only have one ring in them. And I'll explain the significance of that in a second. So we have five different nitrogenous base, right? A, C, G, T in DNA, and U in RNA. Okay, this is very um, important because later on we have to use these things for the rest of the course, basically. So like it's good to spend some time to explain it right now. Now, the adenine, which is what the A stands for, adenine and guanine, that's how you pronounce these things. Adenine and guanine, they are purines because they have two rings. On the other hand, the cytosine, the thymine, and the uracil, they are all pyrimidine and they only have one ring. So on the test, I may ask you, right, which of the following nitrogenous base are purines? Then you have to know is A and G. Right? Everything else is going to be pyrimidine. So how do you remember it? You can try to remember this phrase, okay? It will maybe useful for you. Remember the phrase pure as gold. So purine are A and G. Okay, everything else it's pyrimidine. Okay, pure as gold. Purines are A, the adenine, and G, the guanine. Okay, so on the nucleotides, right? Like these two parts are fixed, but the nitrogenous base you can change it. Okay, some nucleotides will have a uracil on it. Some other might have a guanine on it, right? So there are four different kinds for DNA and for RNA. Are there any questions before I move on?
I have a question.
Yes, please.
Um, can you more clarify ring? Where is the ring?
What is the ring? Yeah.
Yeah, yeah.
So, um, this is a ring, right? A ring is like a closed shape. It could be like hexagon or it could be like a pentagon. But basically, the you see the N here, that's a nitrogen. And then when they don't show it, it's a carbon. So this particular ring, it's like carbon, nitrogen, carbon, carbon, nitrogen. So that's one shape is one ring. So for purine, you have two of these things connected together, and that makes it two rings. That's all it
Okay.
means. Okay.
Yeah. Thank you.
Yeah. What role does the GACTU have in RNA and DNA? Excellent question. I'm going to come back to it in a second. Okay. So let's take a closer look at the DNA structure. The DNA is often described as a double helix. Okay? What is a helix? A helix is basically a spiral, right? But DNA has two spirals that are intertwined together, and it forms a double helix. To help you visualize it, you can think of a rope ladder. Okay, a ladder. Like one that you can climb on, a ladder. And now you take the ladder, and because it's made of rope, it's flexible, you twist it. Okay, once you twist the rope, then you basically get your DNA molecule. Okay, keep thinking about the ladder. The rocks, these are called the rocks, right? Rocks. The rocks that you climb on, that's where the ACTGs are going to be. Whereas the side of the ladder, that's where you will have the sugar phosphate backbone. Okay. So the blue and the yellow ribbon here, that's the side of the ladder. That's the backbone of the DNA, and it's the sugar and the phosphate. Okay. Remember, our nucleotide has the sugar and the phosphate, right? So a sugar, it's going to connect to the phosphate and then connect it to another sugar and a phosphate and sugar and phosphate alternating, and it creates the backbone. Sticking out from the backbone would be the nitrogenous base. Don't worry if you don't fully get it yet. I'll show you another picture in a second. But what's really important in this picture is to notice the base don't randomly pair up with each other. Okay? There are rules that governs what goes with what. If you have a T on one side of the ladder, then it's always going to be a on the other side. So T pairs with A all the time. Yeah, you know what, I'll write A pairs with T. Same thing, right? But it can help you remember it better because AT spells at, okay? And then C is gonna pair with G all the time. Meaning, if you know one side of the DNA strand, you can predict what's on the other side because of this base pairing rule. Okay, I think it's also written on the next slide, but whatever, it's here. Now, I have a question to ask you. Yeah, I mean, you can answer if you want, or you can answer yourself. Uh, A here. What is A? Is a purine or is it a pyrimidine? What do you think? You can look back if you need to.
Pure.
Yeah, if you remember, right? Pure as gold, right? I said. So A is a purine. And how many rings does purine have?
Pure.
Two rings. That's right. Purines has two rings. The way I remember is like purine has two syllables, two rings. Okay. And thymine, hopefully you remember this is a pyrimidine. Pyrimidine. And a pyrimidine has one ring. Okay. So let's take a look at the next one, and hopefully you will understand my point. What about C? Is C pyrimidine or purine?
Pyrimidine.
Pyrimidine, that's right. So also one ring, right? One ring. I'll just put one ring. And then this one is two rings, right? Now, it doesn't have to be pyrimidine on the left side. You can have pyrimidine like on the other side as well. Okay. But if you keep on checking, you will see it's always a purine paired with a pyrimidine all the time. And that is important because it maintains the same width along the length of the DNA. It's always going to be three rings across, right? It's not going to be like sometimes it's like four rings and then sometimes it's three rings, sometimes it's two rings, right? If we do that, then your ladder is going to be like some part is going to be bulging, some part is going to be skinny, right? And then some part is going to bulge out again. And it's just going to be very inconsistent and it makes it very like the structure is not strong, right? And it's gonna break easily. So these base pairing rule ensures the DNA have consistent width along its entire length. Okay? It's always a purine paired with a pyrimidine. It's always one ring plus two rings and therefore three rings across. Are we together? Okay, so what are these ACTGs? Like, what do they mean? We will learn how to decode it in lecture 7. But for now, I actually mentioned last class. Remember the primary structure? You don't have to go back. Okay, just let me do it. Remember the primary structure of the proteins, right? Okay, primary structure is like the order, the sequence, right, of the amino acid. Valines connected to histidine, connected to leucine, so on and so forth. But what determines that? That would be your DNA, right? The ACTG actually are instruction for your cells to read, and they would understand what amino acid needs to go together to build a particular protein. Okay, so that's what the ACTGs are. They are basically a code, right? A recipe for your cell to make proteins. Okay, and we're going to learn how to decode it, right? Later on in the course. If I give you a sequence like this and I ask you to make the protein, you will be able to do it. So here is a more, chemical structure view of the DNA. You can see here is a nucleotide. This is one nucleotide. Okay, one nucleotide because it has the phosphate, it has a sugar, and then it has, in this case, adenine, right? Two rings in it. Here is an other nucleotide, and then another nucleotide, right? Remember, the nucleotides are monomers, so we can connect them together using the same method that we've been doing for the whole unit, for the whole lesson, right? The dehydration synthesis. You can remove a water molecule and then you can connect them together, right? So like I said, over here, circle that again. There is the nucleotide. Let me use a different color here. This over here, the sugar phosphate, sugar phosphate, sugar phosphate. Right. This is called the sugar phosphate backbone. It's written right here. It's written right here as well. And this is like the side of the ladder, so to speak, if you understand the ladder analogy. Okay, so you can see A is going to go with T and C is going to go with G and so on and so forth. What do you notice about this diagram? Like something is kind of strange, I would say, right? Do you notice what it is? No, looks all right to you guys. Everything just
One strand is reversed to others.
Yeah, one side is reversed, it's upside down, right? You can
Yeah.
see this is upside down. So we have two um, words to describe the property of DNA, and they are listed here, right? One of them is complementary. Complementary means knowing one side allows us to predict the other side using the base pairing rule. Okay, so DNA is complementary. You only need to know half. You can always come up with the other half. Anti-parallel means they are parallel to each other. They're always like same width across, but they run in opposite direction. The best way to think about this is like highway. Okay, driving on a highway, the northbound is parallel to the southbound, but then the traffic is running in opposite direction. So that's basically what happens with the DNA. Right? They're parallel, but the direction is opposite.
Oh, this is anti-parallel.
Anti-parallel, yeah, anti.
And yeah,
Yeah, opposite.
anti-parallel.
I mean, they're parallel, but they run in opposite direction. Here, I should
Mhm.
write it down. The two strands are parallel, but run in opposite direction, like northbound, southbound of the highway. Okay. If that doesn't make sense to you, here's another way to think about it. If you think about people lying on the floor, head to toe like this. Okay. The other side would be like this. Also head to toe, but then it's upside down. Okay. Hopefully this makes it better. Okay. And then they will be holding hands with the person next to them. Okay. And that's kind of like the C's bonding with the G's and the A bonding with the T. Now, I know it's already a lot of information, but just one more thing, and then we'll move on to the next picture. What's holding the base pairs together? These dotted lines, these are hydrogen bond. Remember I told you hydrogen bond is going to keep on coming back. Hydrogen bond. If you don't understand what it is, just think of it as like attraction. Okay? Little magnets attracted to each other. So the G will be attracted to the C and the A will be attracted to the T. And you can see in this picture, there are three hydrogen bonds between C and G, but only two hydrogen bonds between A's and T's. So what does that mean, right? Well, it means if you have a strand with lots of A's and T's in it, right? So like that's one strand. And then on the other side, I can predict the other side using base pairing rule. You don't have to write this down, okay? Just watch. Don't write it down. Just watch. Right? So like that's one strand at the top and then another strand at the bottom. This strand would be easier to pull apart compared to something that has a lot of C's and G's in it. Because what's holding it together between the A and T? Only two hydrogen bonds. Compared to if you have a lot of C's and G's, then they have three hydrogen bonds. So they stick together better, right? Right, so knowing how much A and T's is in the strand will allow us to know how much energy is needed to separate it. Now, why do we care? Again, we'll learn more about it later on in the course, but for now, during the pandemic, right? Like how do you know if you have COVID or not? What kind of test do they do on you? There is the one you can do on yourself in your bathroom yourself, right? With the drop pee stuff. And then you check for the lines. But like what's the one they have to send the sample out? What's the test called? It's three letters. What am I talking about? Nobody had it done on you. I don't believe it. They collect a sample from you and then they send it to the lab and you wait a couple of days before they give you the results. PCR? Have you guys heard of PCR before?
Yeah, polymerase chain reaction.
That's right, the PCR reaction, right? So that is for detecting whether someone has COVID or not before they came up with the self-testing kit, right? So when you do these kind of PCR, which is something we'll learn in lecture 9, you have to know the AT contents and the CG contents, okay, in order to run these things. Anyways, don't worry about it if you're not sure what that means just yet. Okay, if I give you a strand like this is one half of the DNA. What will be the missing half? Right? What will be on the other side? Well, that's actually really easy to do. All you have to remember is the base pairing rule. What does A pair with? What did I say a couple slides ago? A always goes with
T.
T. That's you gotta put it right here. A always goes with T. And G always goes with
C.
C. And that's it. You just keep on using the rule to finish the rest of it. I'll give you a minute to fill that out, and then I will write out the answers, and you can compare them, okay? Yeah, that's good. Okay, so C goes with G. TTT would be AAA, ETA. Does anybody need clarification on this? If you don't understand, feel free to ask. No, everybody got it? All right, I will believe you. Thank you. Here's another question. Math question. Oh no, math in biology. Right? It's not too bad. Suppose a cell has 40% adenine. Find the percentage of T, C, and G, right, in their DNA. I forgot to mention this is in the DNA. All right. So here we go. 40% adenine. Well, A always pairs with T. That means if you have 40% adenine, you must also have 40% thymine, right? Because whenever you find an A, you're gonna find a T for sure. So 40% A must be 40% T. If you add these things up, 40 plus 40, this is going to be 80% of all the nucleotides. Which means if you take 100%, subtract 80%, you still have 20% of nucleotides. And the rest will be C and G. 20% are C and G, and they must be evenly split. So 20 divided by 2, it's 10% and 10% because they're the same. All right. That's all. So that's DNA. Let's take a look at RNA. RNA are single-stranded. The backbone is made of ribose as opposed to deoxyribose sugar. And there's the phosphate as well. The nitrogenous base or nitrogen base is ACG, just like DNA, but the RNA have uracil instead of thymine. Now there are three kinds of DNA, three kinds of RNA, okay? Messenger RNA, ribosomal RNA, transfer RNA. Don't worry about what they do at the moment. We will come back to it in the second half of the course. Okay, so that's the RNA. And that's it. Okay. So there are some concept track questions here for you to try. One, two, three. You know what? Let's just do it together. I was gonna ask you to do it yourself, but let's do it together. Why not? First one, which of the following is true? Okay, true regarding purines. Okay, let me launch the poll. We are looking for the true statements regarding purines. Okay, that's all. Um, let's see. Adenines bonding to thymine is stronger than guanines to cytosine. That is not true. I told you between A and T, there are only two bonds, whereas between G and C, there are three, right? So, it's definitely stronger between C and G than it is between A and T. Cytosine is a purine. Nope. Purines are A and G, right? Pure as gold. Purines bind readily to deoxyribose, but not to ribose. So they are saying the purines, the A's and the G's will only bind to DNA, but not RNA. That's not true. A and G are both present in DNA and RNA. Both adenine and thymine are purine. Nope, we know that's not true already. It's supposed to be guanine. Purines have a double ring structure. Right? That's the correct statement. Okay, which of the following is not a feature of DNA? So on the quiz, there will be some questions where they have multiple correct answers, and you have to choose all the right ones to get full marks. So it could be something like this. All right. You will know because the quiz will say the following section, the question will have more than one correct answer. And you will know because you can actually select more than one correct answers. The questions with only one correct answer, it won't allow you to choose more than one. Anyway, we're looking for the correct statement here. Okay, it's been a minute. All right, I'm gonna end the poll. Correct answer. Let's see. Nucleotides within a single strand are connected by covalent bonds. That's correct. Okay, remember I told you, the monomers are always connected together by covalent bonds, right? By removal of water. Next one, adenine forms a base pair with uracil. That's not true. It forms it with T, not U. Complementary strands are held together by hydrogen bonds between phosphate group. That's false. Yes, it's hydrogen bond, but it's not phosphate group. It is nitrogenous base. Complementary strands are anti-parallel running in opposite direction. That is true. Okay, so first one and last one, they are the correct one. And then I told you before, I think some of the questions will be application questions where you have to apply your knowledge, you have to think about it. Is not just like factual recollection, right? So this would be an example of an application question. It's not going to be a lot of those, but they will be there. A chemical reaction occurs slowly at 37 degrees Celsius. When molecule X is added, the reaction speeds up significantly. Which of the following is a monomer of molecule X? Think back to last class. What kind of substance will speed up a chemical reaction? We learn it towards the end. Right? What do we call those things that would speed up a chemical reaction? Anybody?
Enzyme?
Enzyme is correct. That's right. So because molecule X speeds up the reaction significantly, we can reasonably say it is an enzyme. Okay. Now, the question is asking which of the following is a monomer of molecule X? In other words, what's the monomer for an enzyme? So then you got to think about this. What is enzyme? Is enzyme carbohydrates, lipids, proteins, nucleic acid? Which one is it? What are enzyme made up of?
Protein.
Protein is correct, right? We learned this last class, all right? And proteins are made up of amino acid.
Amino acid.
So which one is amino acid? Well, we drew amino acid last time, right? So this is the answer. Okay, so there will be some application questions like these where you have to apply your knowledge, you have to think about it. Um, anyway, that's it. So this is the cutoff for your first quiz. Okay, if you have not already download the lockdown browser and everything, you need to do that before your first quiz, okay? Otherwise, you try to do it on the day off and things don't work out, it's really hard for me to help you out, right? So for practice, you can go to lecture 1. I know I showed you this before, but study resources, self-quiz, okay? Just click on that and do it. Okay, after you click on it, you can start the exam, but before so, you have to download the lockdown browser. Okay, I think it's only going to work on a computer, okay, laptop or computer. It doesn't work on your phone or your iPad. And you don't want to be doing your tests on those tablets, right? Like you need it to be on a device that's big and easy for you to navigate, right? Okay, so this self quiz does not count and you should try it to see how it is, okay? I've checked, only a few of you have done it, okay? So please take a moment to do that. Any
Do
questions?
we have another quiz? I did for first time. I did it, but do we have another under
Yes,
lecture two? Maybe.
exactly, right? So every lecture is gonna have a self quiz, right? So let me just take a look here.
Okay.
How many of you have done it? Um, 7 of you have done it only. Only 7. Okay? And some of you have done it multiple times. That means less than 7 of you have done it. Please, for your sake, do the self quiz, okay? Do it today, do it tomorrow. You have to do it to help you prepare for the test, okay, for the quiz, okay? It's not it's not difficult, but it's not easy, the quiz, okay, the actual quiz. You have to prepare for it. Okay, lecture 2 here, study resources, same thing. There is a self quiz. This one doesn't need a lockdown browser. Okay, and how many of you have done it? One, only one of you have done it. Oh my goodness. Please, 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 please do it. Okay, do it before the actual quiz. When is the actual quiz? The actual quiz is going to be available to you on Monday. That's next Monday, January 27th. The time limit is 15 minutes. Okay. It will be available to you from 6 a.m. in the morning until 11:59 p.m. Okay. You can do it anytime as during that window, as long as you submit it by 11:59. Couple of things to be careful of. Number one, once you start the quiz, the timer will not stop. Okay. Even if you close it and you say, oh, let's study more and come back and do it, it's not going to stop. Okay. Once you start the quiz, the timer will count down by 15 minutes and that's it. Afterwards, you cannot open it again. Number two, do not wait until 11:45 to do it. If you wait until 11:45, you are not going to have 15 minutes because it's due at 11:59, right? So if you wait until 11:45, you will only have 14 minutes to do it. Okay, so give yourself plenty of time. And number three, try to be in a space where you're not going to be interrupted. Okay, like if you have kids at home, try to, do it before they come back from school or do it until after they go to bed or something, right? Try to not get disturbed by it. Like I said, if you run into any technical problem, you don't worry, don't freak out. Just email me and I'll try to sort it out with you. Okay. But this is going to be Monday, and it's going to cover everything on lecture 1 and lecture 2. If you need to check the concept track questions, like the ones we were doing in class, like these poll ones, and you want to like figure out the answers, you can download them. It's right here. Concept track questions answer. Okay? Just download them if you need to. Do we have any question about the quiz? We have class on Monday, okay? Like you still have to come to class during our regular class time. You are not supposed to do the quiz during the class. You do it, within the window outside of class, okay? Maybe between your morning class and my class or afterwards. Questions?
Yeah, I have a question. How
Sure.
can we see the quizzes and test dates? I couldn't see it. I searched it, but I couldn't. Where can I see it?
Yeah, so there are a
Is there any
couple,
option?
yeah, so a couple ways to do it. The easiest way to figure out when things are going on would be go to course information and download the weekly schedule. Okay, so the weekly schedule will tell you exactly when the quizzes are going to be. So your first quiz, lecture 1 and 2,
Oh, okay.
that, okay?
Okay.
Alternatively, you can go to the quiz folders and you see content is available, right? If you mouse over it, it will tell you when it will be available. So you see on the screen, it's January 27th, right? 6 a.m.
Mm-hmm.
Okay, same thing. All the quizzes folders are there. It's not available yet, but the next one is going to be February 10th. Okay, all the
Yeah,
quizzes
I
and tests
saw that.
will be between 6 and 11:59. Okay, on the day.
Now it is locked, so we need one password.
No, it's
Like this.
locked because it's not the quiz day yet, right?
Yeah, yeah.
Yeah, so that's why you cannot click into it. There's no password. On the day, as
Mm.
soon as after 6 a.m., you can click into it.
Oh,
Okay. But like I said, as a practice, you should go to study resources and do the self quiz, right? Because you need to install the software. Have you guys done any assessment in other courses yet, in chemistry, for example?
We
No?
did in math. It was also practice.
Okay, but is there a lockdown
And
browser for math or no?
yeah, and also webcam
Okay, yes.
required.
So for me, for my course, no webcam, right? But you need the lockdown browser. right? If you've done it in math, great, it's the same thing. But still, you need to do the self-quiz. The self-quiz are really good, okay? I Let me just show you, okay? I don't understand why people are not doing it. Maybe you didn't know, or maybe you just didn't get around to it. But it's really good. See, look here. Select all the true statement. It really makes you think about the things that you've learned, right? So uracil are found in DNA only, while thymine found in RNA only. Is that true or false? What do you guys think? We just learned it like for the past 45 minutes. Do you think this is true or not?
False.
False.
False. So you're not gonna choose it, right? And then, the backbone of DNA is made up of alternating purines and pyrimidines. Nope, the backbone is made up of sugar and phosphate, right? So it's like several true or false questions all packed into a single question, and it really will help you prepare for the quiz. Okay? And like I said, it's okay to have a help sheet next to you, but don't have your whole binder in front of you. You won't have time to flip through your notes. You only have what, like 15 minutes, right? So there you go. And these self-quiz you can do as many times as you want to practice before the test. Melissa has a question. If the lockdown browser has no camera, do we still need to show you? No, you don't have to show. I trust that you are you, okay? All right. You don't have to show the ID. You just, do the test as it is. Are we okay? Everybody,
Yes.
yeah, everybody happy, okay? You just have to prepare for it. That's all I'm saying. All right? That's all I'm saying. Okay? Please,
I
please.
think I think all of us, for me, we are still trying to adapt the program and everything. The online it's not easy, so I think that's why we didn't. I did it, but I
Yeah,
think
yeah.
that's why the generally, but we will going to use it,
Absolutely,
get to
right? It's
use it.
a
Yeah,
it's a learning curve, right? And that's why there's so
yeah.
many support and tools to help you prepare for the actual quiz, right? I just don't want you
Mhm.
guys to not know what the platform looks like and what kind of questions to expect and then jump into the quiz on Monday. You will feel overwhelmed that way. Okay, but if you do the practice quizzes, at least you will be familiar with the kind of questions that I will be asking you, right? So these questions that in the self quiz are similar. Some of it is similar to the actual quiz. Okay. Now, I do want to point out one other thing. The quiz that you'll be doing on Monday, in addition to the multiple choice questions, okay, you will also have to do short answer questions for me. Okay, so that this is where you have to type answers. Right. Um, just be mindful of time. You don't have to type a lot, okay? Just answer the questions in a concise manner and you should be okay. Just give me a moment. Let me double check something here. Um, hold on. I built this thing a while ago and I kind of forgot. Yes. Okay. So fortunately, for you, the first quiz has no multi-select questions. Okay. It's just multiple choice with a single correct answer. So you don't have to worry about questions with more than one correct answer. Okay. That's going to come later. Okay. This one, we're just starting. So it's not going to be too overwhelming for you. All right. But you do have to do two short answer questions. Okay. Lastly, one more time. Any questions before we do lecture 3? All right. I'll post an announcement today after class to tell you more about the quiz. I mean, everything is already there, but whatever. I'll post an announcement as well, and I'll post the contact information for the tutor. All right. I think she's from Waterloo, and she can help you guys with biology if you need to. Of course, I'm available too. If you need to send me questions, just email me. I will get back to you. With that said, let's start lecture 3. Lecture 3 is not on quiz number 1. It will be on quiz number 2, though. Okay, so here we go. Lecture 3, cells. Start with some background information. We are, familiar with this diagram, hopefully by this point. It has to do with the organization of life, right? The hierarchy of organization in living things. We spent quite a bit of time in lecture 2 talking about atoms, right? The carbon, hydrogen, oxygen, nitrogen, phosphorus. They can come together to form molecules. Some molecules are small, like H2O only has 3 atoms, but some are big, right? We saw DNA can be very long, proteins can be very long, right? And so they are the macromolecules. And today we're gonna move up the ladder and we'll start to learn about organelles, which are tiny organs. That's literally what it means. Tiny organ, tiny structures that are found inside a cell, and it helps the cell function properly. Okay. And for the remainder of the course, we will be talking about cells. Okay. It's not until the end in lecture 10 where we will talk about tissue. Okay. Between now and lecture 9, we will learn everything there is to learn about the cell. So how big is a cell? Well, this is a good comparison. Okay. If you think about adult, adult would be over a meter tall. If you think about an egg, that would be just about 10 sorry, this is ostrich egg. It's really big. This is 100 millimeter, which is like 10 centimeter. A regular egg would be somewhere between 1 centimeter to 5 centimeter. Depends how big it is. Like quail's eggs are really small, right? But if we keep on going down the scale, we will see cells are not measured in meters. Okay, they are not measured in centimeters or millimeters because they are so small. We measure them in micrometer. Right? We first saw this in lecture 1, right? One, oops, 1 meter is equal to a million micrometer. There's this fancy U here, micrometer. Okay, when we do the microscopy lab, we'll do calculations with micrometer. And inside these cells, some cells are bigger than others, okay, but typically they are within this 10 micrometer to 100 micrometer range. And inside the cells, you would have the organelles. Okay, organelles are about the same size as bacteria, and they're in the 1 micrometer range. And viruses are even smaller, right? So we need a new unit to measure them. They are measured in nanometers. Okay. And when we talk about atoms and proteins and lipids, they are really, really, really small. You can't really see it with even with powerful microscope. So what is a cell? By definition, a cell is defined as the smallest unit that has the properties of life. You know, the properties, the characteristics of life that we talked about, you know, require energy, maintaining homeostasis, order, structure, you know, reproduce, right? So a cell is the smallest thing that will still have all those properties. If you go any smaller than a cell, then they stop having those properties. Okay, if you look at an organelle, it no longer have all those properties, so it's not alive, right? So cell is the smallest unit of living thing, basically. Right? All living things are made of a cell. Doesn't matter if you are a human, a plant, a dog, a shark, a fungus, a mushroom. Everything is made up of a cell if it's a living thing. And cells don't all look the same. Okay, we have over here, these are called Staphylococcus, which is like a chain of bacteria, right? Some of these bacteria can cause things like infections, right? Like besides Staphylococcus, there's also Streptococcus, which cause strep throat, right? So these are balls of bacteria, basically. Okay, this guy over here on the right side, oops, it is called an amoeba. Okay, amoeba is a single cell organism that lives in pond water. Right? Sometimes they can make their way into swimming pools, and then it makes make kids sick. Right? They might have like diarrhea and stuff like that. Right? So that's amoeba. What about this? Anybody know what this picture is showing? What are these things?
Neuron,
Ne
nerve cell
neurons. That's
members.
right. These are neurons. Okay. These are brain cells. Okay. So you see the brain cells here, they have these connection making with other brain cells, right? And so, you know, they look very different compared to the amoeba and compared to the bacteria, right? Even though cells look very different, depending on where you find them, depending on what organism they are part of. They all share some common features. All right. The cells, they all have a cell membrane. Okay? A cell membrane is basically the boundary of a cell, if you would. Okay? It separates the non-living environment with the living environment inside. So that's a cell membrane, which is also known as the plasma membrane. If you remember from last class, the major component of cell membrane is phospholipids, right? Phospholipid. More on that later. Another common feature of cell is that they all have genetic material. Okay? Genetic material would be in the form of like DNA, right? Nucleic acid. Okay? So like, there we go. Some DNA right here. Okay, so that's the genetic material because the genetic material basically is the blueprint for making the cell. Okay? It contains all the instructions that are necessary for the cell to function properly. And finally, there is the cytoplasm. Cytoplasm. The best way to think about this is if you think about a Ziploc bag, you put some water in there and then you zip it up. The Ziploc bag is the plasma membrane, is the cell membrane, and all the water inside, that's basically the cytoplasm. Okay, it's like this, I wouldn't say it's water, but it's kind of like gel-like substance where everything else is going to be floating around inside the cell. Okay? So it doesn't matter what kind of cells you are talking about. Doesn't matter if you're talking about bacteria, neurons, amoeba, they will all have cell membrane, genetic material, and cytoplasm. All right. Let's take a break. When we come back, we will talk about the cell theory. Okay? I will see you in 5 minutes. If you have any questions, just leave it in the chat. And we are back for part 2. Let's start with the cell theory. So the cell theory has four parts to it, not three, 4 parts to it. And the first part goes like this. Every living organism consists of one or more cells. Well, we kind of know this already, right? But what you might not know is the organism can be as little as one cell. Right? So here we have this thing called the paramecium. It's in the same category as the amoeba I showed you before. These are living in like fresh water usually. Um, they're not bacteria or anything like that, okay? Like in terms of organisms, we can like we have animals, so that's what we belong to, animals, plants, and then there is other groups as well. Okay, so one of the group is called protists. You don't really have to know this. I'm just showing you in case you're wondering. Okay, protists. So under protists, you have things like amoeba and paramecium. Okay, so they can be as small as a single cell. Single cell. All right. We have something called C. elegans. It's this worm thing that scientists know a lot about. It's actually what they call model organisms. They use it for research as a starting point for many things like medicines, genetic engineering, so on and so forth. And the whole thing is made up of about 3,000 cells. Okay, C. elegans. Humans, we have trillions of cells, okay? 37.2 trillion cells, okay? Probably more if you're bigger and fewer if you're smaller, okay? But, it's a lot of cells, okay? So individually, each cell has their own dedicated job, but they all work cohesively for the well-being of the entire organism. So that's the first part of the cell theory. Next part. A cell is the smallest unit of life. We know that already. Individually alive, even as part of a multicellular organism. Did you know if you collect cells from your body and you put them in a dish, this is called Petri dish, and then you supply them with some nutrients, that's what this red fluid is. You supply them with the nutrients they need to survive, and then you put it in an incubator that's 37 degrees. your cells will be able to live outside of the body for some time. They will eventually die, but they are able to live out of the body as long as you supply them with nutrients and the right temperature and the right pH and all that stuff. So individually, they're alive. They don't need to be part of you in order to survive. Okay? Of course, you need to have the right condition. All right? Now, there is something called a HeLa cells. That's what they look like. And again, anytime I show you pretty pictures like these of cells and organelles, keep in mind they are not the natural color. Okay? The natural color is just transparent. Everything is super thin. And if you look at them, you just see right through them. Yeah. But we can use dyes and fluorescence to color different parts of a cell to highlight them. In any case, these are HeLa cells. So what are HeLa cells? HeLa cells are very useful in modern research. Okay, you can buy HeLa cells online. I'll show you right here. The company, many companies sells them. Okay, but you can buy 20 microliter. Okay, that's a microliter is a millionth of a liter, right? So 20 microliters, you can hardly see it in the vial, and it's gonna cost you $486. Okay. Now, why are they so expensive? Well, part of the reason is you only have to buy them once, and you can keep on growing them, right? They don't stop growing, right? And for that reason, because you can grow them easily and then you can manipulate them easily, HeLa cells are very useful for doing research on a wide range of fields, okay, including cancer treatment, vaccination, okay. You can grow poliovirus in them and then try to develop a polio vaccine using these HeLa cells, and it's just very commonly used, okay. So where did the HeLa cell came from? Well, the HeLa cell came from this person, this lady. Yeah, her name was Henrietta Lacks. Okay, and that's why it's named after her. Okay, HG is the first two letters of her first name, and LA is the first two letters of her last name. Okay, so she had cervical cancer, and when they took a sample from her, a biopsy, right? It's called. They try to grow the cancer cell in the dish, in one of these dishes that I showed you, and to their surprise, the cells does not die. Okay, like if we take our normal cells and try to grow it in a dish, even if you give them optimal condition, they won't survive forever. Okay, like after some time, they will all die. Right? But not the HeLa cells, not the cancer cells. If you keep on feeding them with nutrients, they could keep on dividing and dividing and dividing. When the plate gets full, you can take a few cells, put it in another dish, and then they will just grow and grow and grow again, right? So this immortality is a property of cancer cells in general. And so what happens is, that became a cell line, right? And the company started to sell these cell lines for people for research. And as I show you, they are quite expensive, right? So they make quite a bit of profit from selling the HeLa cells. But eventually, Henrietta died, and her estate and her family are suing the company, right, Thermo Fisher, that was selling her cell line, right? They're saying they didn't get her consent to essentially profiting from her cells. Okay, and there was some back and forth between the lawsuit, but ultimately, the company settled with the family. Okay, and I forgot how much they paid them, but it's highlights the importance of getting consent and ongoing consent, right? Something we discussed in lecture one. All right, so that's part two of the cell theory, right? The cells are individually alive, right? They can live outside of your body.
Sorry, did they sell her cells?
They did, yeah. They sell her cells, okay? And because they are
Oh.
cancer cell, they can keep on growing them, right? Remember I was telling you, right?
Mm-hmm.
If the play is full, you just put them in a new play and then you grow them. You can actually freeze them down, keep them at -80, and when you buy it from the company, they ship it to you in dry ice, and you get a little tube with the cells in it. And you're supposed to grow it in your own dish, and then you make more yourself, right?
So since that day,
Yeah.
that it's growing till
Yes,
now?
That's right.
Oh
That's
my
right.
gosh.
Yeah. So like they have many copies of it, but ultimately all of them
Oh,
originate from that original sample. Okay.
oh my. Okay.
And we will learn more about cancers later, but one of the properties of cancer is um, immortality, right? They keep on growing and growing. They just don't die. That's why it's so hard to treat them. Okay, next part of the cell theory. All living cells come from division of pre-existing cell. We don't really know where the first cell came from, but we know to get more cells, a cell needs to divide. Okay, so like you would start with one cell, and then the cell would split, and then you would get two cells. Okay, and then these cells would split again, and you would get four cells. So that's how we grow. Okay, um, when you're a baby, you have fewer cells than when you are in an adult. Where do all those cells came from, right? It came from the division of pre-existing cell. What about the very first cell? Well, you and me came from a single cell, and that single cell was created from the sperm and the egg coming together. And from that single cell, divide, 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 and then it becomes us, right? So all living cell comes from division of pre-existing cell. You are looking at onion cells here. Again, they are stained to make it more prominent, the colors. And you can see this is an onion cell that's undergoing division. It's about to split, and then there will be a wall that forms afterwards, and now you have two cells. Okay, we will learn this in the second half of the course. Finally, last part of the cell theory, cells contain hereditary material which are passed on during cell division. Okay? So I told you we have 46 chromosomes, 46 DNA materials, and when the cell divides, okay, each of them will get 46, and it will be an identical copy of the original. Okay? So those are the four parts of the cell theory. Does anybody have any question? Very well. How do we study cells? Well, they are really small, right? Like if you look at your hand, it's not like I can see the cells that makes up the hand, right? You need to have a microscope to see better, right? To magnify them. And the first microscope was invented by a Dutch scientist, but he wasn't really a scientist. He was more like a tailor, and he's just doing this on the side as a hobby. But then he got so good at it, then eventually people call him like a scientist. Okay, so his name was Antony van Leeuwenhoek. That's how you pronounce his name, Leeuwenhoek. Okay, so he was obsessed with trying to make a lens and then put it onto this handheld device. And he was just observing rainwater, okay, and water that he got from his plant pots and stuff like that. And when he looked at it through this handheld device that he made, he discovered a whole world that was unseen by the naked eye. Right? In the pond water, in the rainwater that he was observing, he saw all these living things are moving around. Okay, he called them animalcules. Okay, so like they are molecules, but they are like animals. So he called them animalcules. Okay, and he tried to publish his findings. So he actually do drawings um, of the things he saw. And then other people didn't believe him, right? He thought he was just crazy, right? He's just very good imagination, right? And then people tried to recreate the lens, but they didn't have the craftsmanship to do it. So no one was able to reproduce his result, and people didn't believe him. And when someone want to come and use his instrument, he worried that his instrument is going to get copied by others, so he refused to show them, which doesn't really help his case, right? Like if you want people to believe you, then maybe show them what you are seeing, right? But eventually, long story short, he allowed people of high stature in the community at the time to come and verify his results. Okay, so like there were some lawyers, there were some officials, government officials, and they came and they saw what he saw, uh, and that's basically the start of the microscope era and it leads to a whole field of, studying cells, discovering bacteria, and all those things. Okay. So nowadays we use these microscope, and this is a light microscope. It's the one we actually have at school. They're not too expensive, roughly $400 for each, which is reasonable price for a microscope. Okay, we will have a microscopy lab later on in the course. There are high power microscope that cost up to millions of dollars. If you get the whole setup, this is the microscope, and then with the computers and the software and everything, it costs over $1 million. And people use them for diagnostic purposes at the hospital. They might use it in research lab. Okay, compared to 400, it's nothing, right? The $400 microscope, they can magnify up to 400 times or even 1,000 times. For these very powerful microscope, they can magnify up to 2.5 million times. Okay, 2.5 million times. So you can see here in this picture, it is the distance from here to here is 1 micrometer, and then you can see the structure of the cells vary in a lot of great details. Okay, so wild guests here. I think some of you know, based on how some of you answered the question, I feel like some of you have taken science before, which is great. Okay, but if you have to guess, if you have no idea what this is, I'll tell you it's in the body somewhere. Do you want to know which part of the body this might be from?
These are cilia.
Yeah, these are cilia. That's right, right? So you probably have studied them before. Cilia are little hairs that are on the cell, and you would find them in your airway, for example. Okay, when we breathe in all the dust particles, the cilia is going to help sweep them up to your throat, and you have two options. You can spit it out or you can swallow it, right? There is an other place you will find these little hair in the body, in the female reproductive tract. Okay, men don't have it, but in the female reproductive tract, the fallopian tube is called, or the oviduct, where the egg is going to be traveling along. The hair will help guide the eggs to the uterus. Okay, and that's what they look like, these little cilia, and you can see them in great details with these high power microscope. Okay. All right, time for a poll questions. Try to do the polls if you can, please. If you don't see the poll, feel free to just type the answer in the chat. The key is to participate. All right, so which of the following is the concept of the cell theory? Correct answer is D. A cell is a basic structural and functional unit of living things. Not too difficult. This one, I'll do this one because I haven't really talked about it completely yet. All of the following are present in all cells, except, so if you go back to our earlier slide, you will see we have a checklist here. All cells have cell membrane, genetic material, and cytoplasm. So the only thing on the list that's not there is the nucleus, right? I haven't teach you about the nucleus yet, so I will in a bit. All right, new cell membrane and nucleus. So when it comes to cell, we try to categorize them into different types. Okay, cells. We have the more primitive type of cells, which are called prokaryotes. Okay, these are P for primitive, they're simpler. Okay, and then we have the more complicated ones called eukaryotes. Sometimes it's okay to say eukaryotes or eukaryotic cells, prokaryotes or prokaryotic cells. Okay, so these are simpler ones. These are more complicated, which is what is written here, right? Eukaryotic cells are much more complex and are generally larger than prokaryotic cells. In eukaryotic cells, the biggest difference between eukaryote and prokaryote is the presence of something called the nucleus. Okay? So in the center of the eukaryotic cells, you would see this big spherical structure called the nucleus, and that's where you would keep all your genetic material. Okay? It's safely protected inside the nucleus. Prokaryotes don't have that. They have genetic material, but they're not enclosed within the nucleus. Eukaryotic cells have many different types of organelles. So organelles are these tiny organs, tiny structures inside the cell that has different functions. So we can have the mitochondria here, the Golgi body, so on and so forth. We will be learning the functions of those in this lecture. So what are examples of eukaryotic cells? Animal cells, plant cells are examples of eukaryotes. You know, those protists that I told you about, protists are also eukaryotes. If you forgot what protists are, those are the amoeba that I showed you, right? The paramecium, and all the stuff that Leeuwenhoek, the guy with the handheld microscope, when he was looking at the uh, rainwater from the gutter or from his plants, right? Whatever he was seeing, those are protists as well. Okay. Kelp is an example of a protist. Right? If you need something more concrete to understand what a protist is, okay, kelp. So we are made up of eukaryotic cells. Okay, our cells have a nucleus in it. Although there are some exceptions, but I will tell you that next semester. Prokaryotic cells, they are smaller, simpler, do not have a nucleus, and there are a few types of organelles in them. Okay. Bacteria are prokaryotes. Okay. I don't know why I put the word most here. Uh, all bacteria are prokaryotes. Okay. There we go. Bacteria are prokaryotes. Do you guys know any bacteria like from the news or something? Like usually they would say, oh, throw away your eggs because of something something contamination. Don't eat the salad because of something something contamination.
Salmonella,
Salmonella, that's right. Salmonella is a type of bacteria. Anything else besides Salmonella?
E. coli,
Yeah,
Escherichia coli.
that's right. E. coli. Right. So these are all examples of prokaryotes. Okay. Listeria is another big one. They usually contaminate like cold cuts, right, and stuff like that. All right. Cell membrane. Let's take a look at the cell membrane here. The cell membrane, which is also known as the plasma membrane, surrounds all cells. Doesn't matter if you're eukaryote or prokaryote, you're gonna have to have a cell membrane to separate the inside from the outside, right? Otherwise, everything's just spill all over the place, right? The cell membrane function as a selectively permeable barrier. What that means is, oh my goodness, what that means is it regulates what can go in or out of the cell. Okay? It's not free for all. It's not like anything can come in and anything can leave the cell. There are regulation in place. You, for example, don't want the nutrients to leave the cell, right? So you want a way to bring them in but keep them in the cell. You don't want harmful toxins to be coming into the cell. but you do want a way to excrete toxins out of the cell, right? So the cell membrane plays a role in that. It regulates what can cross the membrane and gain access to the cell. Okay. We will learn more about the cell membrane in lecture 4. Right? So what is the cell membrane made up of? As you might remember, the cell membrane is made up of primarily phospholipids, and you have two layers of it. That's why it's called phospholipid bilayer, and it's arranged like this, like a sandwich. Okay? The phospholipid has a hydrophilic head and hydrophobic tails, and it forms a sandwich like that. Okay? So the hydrophilic head is either facing the water outside or the cytoplasm inside. Cytoplasm is mostly water anyways. Okay, it's a gel-like substance, but it's made up of water primarily. And you have a thick layer of hydrophobic tails in between. Okay, so this hydrophobic tail basically forms a barrier for substances. Okay, kind of blocks them. Okay, so we'll talk more about what actually can cross and what would have a difficult time doing so. This is a real picture of the cell membrane using one of those high power microscope, right? The cell membrane is just this thing right here. This is the cytoplasm. This is the outside. And the cell membrane, you can see there are two darker lines with a white gap in the middle. The white gap is where the tails are. And the two dark black lines are the hydrophilic heads. Okay. That's it for the PowerPoints for now. I do want to take the next 20 minutes and draw a picture with you. Uh, it will help you understand the organelles easier. When you draw it out, it usually will be easier for you to understand. Some color would be nice. If everything is in black and white, it's really hard for you to follow. And you want to draw this big, right? So take out a new piece of paper. Okay, we're gonna start here, and then we're gonna come all the way out here, and then later on we're gonna draw more here, and then we're gonna write some notes here, right? So you're planning ahead. Okay? I always like to tell people what the note is gonna look like, so they don't run out of space. Can I move this guy over there? Never mind. I'll just do it here. Oh, did I move it? I did. Okay, there we go. Do I have one that's done? Let me see. Yeah, this is what we're going to be drawing, okay? This is what we're going to be drawing, this whole thing, okay? And you're going to fit all that into your notes. So here we go. All right, we are going to be drawing a eukaryotic cell and some of the key components. Okay, so we are made up of eukaryotic cells. So here is the cell membrane, and then here we have the nucleus. And what we're gonna do is we're gonna take the nucleus out, okay, and then we're gonna be enlarging it and try to put more details into this thing. Okay, so here we go. So this is the nucleus. And I'm going to draw it a little bit 3D. I'm going to open up the nucleus and take a look at the inside. So, it's like as if I cut out a chunk of it like this. Okay. It's okay if your diagram is not, super pretty. It doesn't really matter. You can reorganize it after. And stop me if I'm going too fast. Now, the nucleus is actually covered by an envelope, by a membrane. All right? So that's what this purple coating is. There we go. So that is what we call the nuclear envelope. I'm gonna put it here for you. Nuclear envelope. The nuclear envelope surrounds the nucleus. Okay. It's supposed to cover the whole thing, but remember, I cut it open for you to see the inside. That's why I didn't cover the whole thing. Right? It covers the nucleus. And it's important to cover the nucleus, obviously, because that's where you keep your genetic materials. You don't want it to be, leaking all over the place, for example. So on the nuclear envelope, you will have some pores, some openings. Right? These openings, they are called nuclear pores. Okay, nuclear pore, just one or pores. And they are all over the place on the nuclear envelope. And the function of the nuclear pores are to allow things, I'll tell you what things later, to move in and out of the nucleus. Okay. I'm just going to move it over here, give myself more space. Nuclear pores. Just stop me if you don't understand what am I doing or if you need clarification. It's no problem at all. We good? Can draw more? All right. Inside the nucleus, you have another spherical looking thing. Right? There's going to be another spherical looking thing right here. I'm just going to show a little bit of it. I will give you an analogy in a bit to help you understand this. Okay? So this inside thing that's inside the nucleus is called the nucleolus. Nucleolus. And the nucleolus, one of the functions of it, is to make another organelle called ribosome. Never mind what that is for the moment. We will come back to it. But that's what it does. It makes these things called ribosome. Okay, so what's in this area here inside the nucleus? Well, that's where you're gonna find your genetic material, okay? That's where you're gonna find your genetic materials. So we should put a few threads there. And I want you to understand these genetic materials, which of course are DNA, uh, they are actually very, very long. Okay. DNA, like each cell has over 2 meters of DNA, all packed into a structure that's, a couple of micrometers big. Right. So how do you do that? Well, it exists in the form of what's called chromatin. Chromatin. Right. So let me try to explain this to you over here, and hopefully it will make sense. So here we go. We learned about DNA today at the beginning. DNA is a double helix like this, and then, the lines I'm drawing, those are the ACTGs, right? So this is called naked DNA. It's just on its own. And like I said, you got 2 meters of this stuff that you need to pack into a tiny, tiny nucleus. So how do you do it? Well, the DNA actually needs to wound themselves up around proteins. Okay, they will wound themselves up around proteins. Okay, so there is going to be like a protein right here in the middle. Okay, and the DNA just kind of goes around them. There is going to be another protein here, another protein here. Okay, the easiest way to visualize this is to think of a yo-yo. Okay, you know yo-yo, guys? Let me see if I can find a picture of a yo-yo. Okay, like these things, right? These yo-yo. Okay, so the rope is like the DNA, and I don't know what you call this thing, the the circle. That's the protein. Okay, and then you will wrap the DNA around the protein, and then when you run out of space, you're gonna loop around another a yo-yo, and then you're gonna wrap it around it, and so on and so forth, and it's gonna help you package the DNA basically. Okay, so these are packaging proteins. Okay, they have a name, but never mind. We don't worry about it for now. So when you loop the DNA around a protein, then basically in this form we call it chromatin. Okay, chromatin is essentially DNA wrapped around proteins. Okay, and that's the form that it exists inside the nucleus. However, when a cell divides, you need to pack up the DNA even more so it's easier to move them around. Okay? Just like if you have to move from one apartment to another apartment, you gotta pack your stuff into the suitcase and then makes it easier to move them around. So it's gonna pack up with even more proteins, right? And it's gonna allow them to kind of loop up like this, loop, 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 loop. Right? And it's gonna be very, very condensed, very, very condensed. Yeah, you can do whatever shape you want. Okay. And usually they would double up, right, and make copies of themselves, which we'll talk more about later. Okay, but then you would get this very condensed version of the DNA with lots of packaging protein, and that's what we call a chromosome. Okay, so chromosome it's basically same as chromatin, but chromosomes are more compact together. It's more condensed compared to the chromatin. So in the nucleus, you have the chromatin. It's only when you need to copy the DNA and do cell division, then you got to change it into chromosome. Okay? Excuse me. Any questions about anything that's on the screen? Really?
Oh, chromatins has only DNA and proteins, right? That's all.
Yeah, and so
Okay.
is chromosome. Chromosome too, but it's just like more packaging protein, so you can like condense it even further.
Mm-hmm. Okay.
All right, I'm just gonna give you one analogy to help you understand this. You don't have to draw it. And then I will call it a day, okay? Don't draw this. It's just, extra things to help you understand. I want you to think of, I want you to think of an avocado. Right, avocado. Of course, the avocado is going to have a seed in the middle. Okay, and then we're gonna have the You don't have to draw this, okay? Really, just listen to me ramble on, and hopefully it'll make sense. And then we have the meat here. Avocado, okay? You know avocado, right? So think of the avocado as the nucleus. The skin, what would the skin be like? The skin of the avocado is like which part here? Hopefully, you will know this is the nuclear envelope. Okay. And the seed, and the one that you don't eat, you take the knife, chop it, and then turn and take it out, the seed, that is the nucleolus. That is like the nucleolus, okay? Like if this was a nucleus, then these different parts would be like what we're trying to draw here, right? And then the avocado, that's where you will find the chromatin, okay? Location of chromatin. Right? They're like gonna be like here, right? Da, 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 da. And finally, if you take a toothpick or a fork and then you poke a bunch of holes on the skin, right? And those would be the nuclear pores, right? I hope that's better. Right? And that's all we're gonna learn for now. We're gonna add more to this picture. Still have a lot of details to include. So you might want to reorganize it a little bit afterwards. All right.
I have a question.
Mm-hmm.
Naked DNA and protein make chromatin, and many chromatin together make chromosome.
How do let's say, okay, so chromatin and chromosome are basically same, but it's compact even more, right? So sewing threads, right? So sewing threads, yeah. How do they come in the package? They wrap them around a spool, right? With me so far?
Yeah.
That's chromatin. But if I take a bunch of these things, right, and then put it together into a bundle, right, like into a bigger box or something, then that's like chromosome, right? It's like the same thing, but like more compact, more together. Is that better or I don't know.
I have a question. Is
Sure.
nucleus is the important, the most important thing in the cell, right?
I
Is
wouldn't say
it?
it's the most important. All of them are kind of important, right? But yes, the nucleus you cannot live without. Okay. But
Oh,
that question is like, would you say, the kidney is the most important organ in the body? Well, you cannot live without a kidney, but you also cannot live without your heart, right? So we cannot really say it's the most important, but it is important, right? But with
mhm.
all the organelles, each one has their own role, and you kind of cannot live without one or the other, right?
Yeah, because as I remember in my back home, my teacher called the nucleus as a cell's brain.
Yeah, because it contains the DNA, right? The DNA kind of
Mm,
controls what everything else is going on in the cell, right?
okay.
There's an other question. Would you say that the chromosomes are in the DNA or is a collection of a lot of DNA? The chromosome is the DNA, okay? But it's just really packed together, really wound up together, okay? So chromatin, chromosome, they are all DNA plus a bit of packaging protein, okay?
In this picture, avocado, whole thing, green thing is chromosome.
The meat is where you will find the chromosome and
Yes.
where you will find chromatin, actually, chromatin, right?
Mm.
It's chromatin, and it's only chromosome when you try to divide a cell. All right.
Chromosomic is the packed of chromatin.
Chromosome is packaged chromatin. Is that what you said?
Yeah,
Yeah, sure. It is. Yeah.
yeah,
Very
yeah.
good.
Okay.
Can we say like lots of chromatin is together and it becomes a chromosome?
It's not about a lot of it. It's just about I don't know. Let me try another analogy. It's really difficult, I suppose. Okay, this is chromatin, okay? Now you crunch it up, then it's chromosome. You see
Oh,
that?
okay, okay.
Yeah,
Like
it's the
it's
same thing,
stick together.
but it's just really packed together, really compact together, right? Okay.
All right.
One last analogy. I just thought of this. Instant noodle, you know instant noodles, right? When it's dry, it's
Mm-hmm.
all packed together, right? Okay. But once you put it in
Yeah.
water, it all opens up and goes all over the place, right? It's not as compact, right? It's the same thing before and after. The only difference is whether they are packed or whether they are loose, right? That's
Mm-hmm.
chromatin and chromosome.
Okay.
Don't get too hung up on it. It's gonna be okay eventually. Does all human cells have nucleolus? Yes, if the cell has a nucleus, then it will have a nucleolus. The nucleolus is always inside the nucleus. You cannot have a nucleus without a nucleolus. Okay, so that's that. Very good questions. Anyone else? Okay, so we do have class next Monday. I have to emphasize that. Okay, but you will also have to do the quiz as well. All right, study hard. If you run into any problems, with the study guides or things you don't understand, just email me. My email is on the first slide of every PowerPoint deck, and I will help you accordingly. Thank you, guys. Stay warm. Have a good rest of your week. Bye bye.
Thank you, Vincent.
Yeah, no
Take
problem.
care.